Hi and Assalamualaikum. My name is Nurul Fatin Ashikin binti Abdul Razak with number matric S58239. Today, I want to present my FYP proposal. This is my table of content that include 4 chapters, references, and Gantt chart. For 1.1 is introductions of nanomaterials. Nanomaterials are being considered to be fundamental building blocks of nanoscience and nanotechnology. Nanostructure science and technology is a broad and interdisciplinary field of research and development that has made it popular over the last few years all over the world. It has the potential to transform the way materials and products are made, as well as the variety and kind of functions available. It already has a large commercial influence, which will very certainly grow in the future. DNA is examples of nanomaterials that contain 2 to 12 nanometers. For 2.2 green synthesis of nanomaterials, the synthesis of diverse metal nanoparticles utilizing bioactive agents such as plant materials, microbes, and various bio-waste such as vegetable waste, fruit peel, trash, eggshell, agricultural waste, and so on is known as green synthesis of nanomaterials. Biological nanoparticle synthesis is a more convenient, cost-effective, and ecologically friendly method than traditional chemical or physical method. As a result, biological processes are frequently referred to a green synthesis. 1.3 Problem Statement. The potentials of bacteria to develop antibiotic resistance has been a global issue due to their ability. There are many discussions have been made on the approaches to deal with antibiotic resistant problem. Therefore, finding for safe and effective antibacterial agent also has been encouraged. Furthermore, the antibacterial activities is depend not only on the nanoparticle size, shape, chemical composition, and surface features, but also on the bacterial species. There are several ways to obtain magnesium oxide nanoparticle, such as biological method for the synthesis of nanomaterial that will be the alternative to chemical or physical method. Chemical method is not really good for human and living things. That's why we have to overcome the problem by using green synthesis without toxic chemicals that bring harmful to us. Next, 1.4 Significance of study. The magnesium oxide nanoparticles were synthesized using a local plant, lemongrass leaf, because they also possess good antibacterial, anti-cancer, and antioxidant properties. It is also easy to obtain and more cost-effective. Furthermore, the antimicrobial properties of the magnesium oxide nanoparticles serve well as an alternate antibacterial activities agent, gram-positive and gram-negative. Presumably due to the differences in cell membrane structure between organisms. Last subtopic for chapter 1 is 1.5 Research objective, such as to synthesize magnesium oxide nanoparticles via green synthesis brought using lemongrass leaf extract. Secondly, to characterize the synthesis magnesium oxide nanoparticle using ultraviolet visible spectroscopy, Fourier transform infrared spectroscopy, X-ray diffraction analysis, scanning electron microscope, and thermal gravimetric analysis. Lastly, to apply the characterized magnesium oxide nanoparticle in antibacterial study against E. coli and S. aureus. For chapter 2, literature review, 2.1 is magnesium oxide nanoparticle. Magnesium oxide nanoparticle with average size 20 nanometer. Other than that, it was confirmed by visible color change from colorless to dark brown. 2.1.1 Green synthesis of magnesium oxide nanoparticle from lemongrass. Plants are most frequent biological substrate for magnesium oxide nanoparticles because they have a cost-effective and easy to process. That is figured to illustrate the process. 2.2 Lemongrass, known as Cymbopogon cibratus. It exhibits a wide range of pharmacological. Lemongrass is a tall and fast-growing. 2.2.1 Phytochemicals of lemongrass. Phytochemicals are the chemicals compound which are produced by plant. The table shows solvent system use. 2.3 Characterizations of magnesium oxide nanoparticles. 2.3.1 by UVP spectroscopy. Next, FTIR, XRD, SEM, and TGA. That spectrums show result of magnesium oxide nanoparticles of rosemary. For 2.4 is magnesium oxide nanoparticle as antibacterial study. There are previous study using agar-well diffusion method. For chapter 3, methodology, 3.1 is about apparatus such as lemongrass filter, pepper, magnesium nitrate, sodium carbonate, deionized water, beaker, magnetic stir spatula, glass rod, petri dish, and measuring cylinder. 3.2 2. Show the lemongrass extraction process from lemongrass leaf to samples of powder. 3.2.1 is phytochemical tests such as test for flavonoid and phenols. 3.3 is green synthesis of magnesium oxide nanoparticle. The flow chart shows synthesis of magnesium oxide nanoparticle process. 3.4 is characterizations of instruments such as UV-Vis, FTIR, XRD, SEMS, and TGA. Lastly, Chapter 4, expected result. We expected that we have a better result by using magnesium oxide nanoparticle with lemongrass extraction. So we can get a good inhibition zone in antibacterial study. So this is references and Gantt chart. Thank you.